It's a great pleasure for me to be here talking about some of the research projects we are doing in Milan on nano system for selective drug delivery. Before we start with the main topic today, so the application in central nervous system, just a brief overview of what I'm doing in my lab in Milan. My lab is dedicated to the development of proper formulation for the dimensional network and colloid-based material, mainly organic, but also inorganic, especially in the case of inorganic nanoparticles, but mostly organic. So we start from monomers or from polymers already formed, can come from synthetic sources or natural sources. As you know, there are both advantage and disadvantage of both of them. Then we can introduce post-polymer functionalization that we will see in this talk today. They can be used for tracking, for cell attachment, and that also give very strange and results to give also other properties that we are not planning to have, and we will see in the next slide. Then formulated like hydrogel or nanoparticle, nanogel, colloids in general, and then these are the main field of application: agriculture, medicine, sensory, and water treatment. As you know, like in every academic lab, most of them are changing, while medicine is from the beginning, from my PhD, so it's the main topic that I'm interested in. In the field of medicine, I'm interested in the development of biomaterials in terms of responsive nanoparticles or nanogel. Today we speak about both of them, but also to the dimensional network, even if they're not part of the topics, or if you're interested in or for collaboration and everything, is something that we are also doing. So mainly hydrogels, so three-dimensional network with water, or B-gels, the continuous structure when you have hydrophobic and hydrophilic domain with the same volume, but it's not an emulsion, so both of the two phases are continuous phases and not one dispersed and the other continuous. And also, with also some experimental micro robots and the possibility to coat them with gels for drug delivery. But today we talk about selective drug delivery. But before we discuss the medical issue, like that, what we in my lab are focusing, as I told you a couple of slides before, a lot of attention is dedicated to post-polymer functionalization. So we are interested in introduce functionality after the polymer formation. Why? It is true that we are adding an extra step with respect to the synthesis, but adding a functionalization before the polymerization can present many issues because they can be an obstacle to the polymerization. They can also react during polymerization. So mainly what is done in the next step. Now they are commonly known under the name of click, that also received the Nobel Prize a couple of years ago. And mainly we can, attach some moiety that can be fixed or mobile, depending on the application we are thinking about image, due to the event of image, cell attachment, drug delivery, response. That's we will see some example in the next slide. We are interested, as you know, in developing controlled drug delivery. You know very well what I'm talking about, the possibility to avoid under and overdosing and maintain a drug level within a desired range. Even if today we are talking about nanoparticles, so our main interest is not on the kinetics. That is more interesting, for example, if we are developing the formulation of a transdermal patch or an oral pills. We are also interested to deliver the drug within specific cells without an uncontrolled release in the other environment, so in the selectivity more than on the kinetic. The kinetics is important because they need to carry the drug inside and not outside, but not for the many issues. So when we are talking about drug delivery, we can be interested in nanodrug delivery system, in many properties. They can have enhanced penetration, controllable release, extend circulation, increase uptake, overcome the medical drug resistance. We can attach both of them with different sides. We will see how we'll do after we finish the introduction section. We move to the specific medical problems. About nanoparticle design, you know very well that there are different kind of strategy we can work with in order to tune and design the proper nanoparticle, and all these and all of them affect the property have, the interaction with the biological environment, and so on. So we can start, for example, from the material. The most common, we can decide to choose if they're inorganic. If they're organic, they can be dendrimer, can be liposomes, extracellular vesicles, classic emulsion, or nanogels. And this is depending on the material, but you can also have other strategy in order to tune all the properties we see in the previous slide. So the size, for example, that depending on the medical need can be more strict or not. We can change the shape, can be sphere, cube, rod, star, whatever we kind of like. And this shape is not a matter of fashion, but it is related to the specific application they can have, the interaction with the biological environment, and so on. Another possibility is also to decorate the surface, as we said, with the functionalization we said before. The surface decoration can work very well if, for example, the most common use is pegylation, that is mostly used in order to have a stable colloid and avoid degradation and the phase separation, but there are also other kind of functionalization that can not only work on the physical stability of the nanoparticle, but also can tune the drug delivery, can tune the selectivity, as we will see in the next slide. After this brief introduction of what we are talking about, we start today. Today, I would like to show you the results we obtained, some of the results we obtained in the last more than 10 years on spinal cord injury in a collaboration with Mario Negri Institute for Pharmacological Research in Milan, that is responsible of the in vitro and in vivo studies on the biological studies. As I told you from the beginning, I'm chemical engineer by training, so I'm not a medical doctor, I'm not a biologist. I learned something, but not that much. I have to be honest with you on that, with a real multidisciplinary project. So what about spinal cord injury? Spinal cord injury is a dramatic condition that results from a primary event. This primary event is a mechanical event. So it's not something genetically, something that takes place after an accident, a motorbike accident, a car accident, and the use of gun for a military field, for example, whatever. That is then followed by the secondary injury. This one is the main cause of the post-traumatic neurodegeneration. In European Union, for example, every year 10,000 people suffer from spinal cord injury, and almost all of them leave permanent neurological consequences. Nowadays, there are no solving therapies available, but the research community is pointing toward the use of biomaterial to provide in situ drug or cell delivery. Today, in my lab, we are working on both sides. Today, we focus our attention on drug delivery. The peculiarity of this is that it is something that has a specific location within the body. So there's a specific target where the accident, where the mechanical injury takes place to treat. This is why it's something that works very well with biomaterials. So basically, starting from primary event, so synthetic or nitric oxide, oxidative stress, microglia activation, local inflammation, they can cause many events like neural necrosis, neural apoptosis, current formation, demyelination, and disruption of morphofunction F pathways. So on one side, the main aim is to have neuroprotection to counteract the mechanism of secondary injury and to minimize the pathological consequences, while neurodegeneration tries to rebuild the damaged tissue. So basically, what is done after the primary injury is that you can see different actors that take place during in this field. The actors are the cells that are responsible of all the interactions. So we are the infiltrating macrophage that after mechanical spinal cord injury obtain different kind of phenotype, astrocyte neuron, and all the classic cells present, of course, in the central nervous system. So basically, there are no solving therapies present, in the field, and we have some sort of limitation, and our strategy is to neuroprotect, to counteract the mechanism of secondary injury and minimize the pathological consequences, and on the other side, trying to rebuild the damaged tissue to obtain a multi-target therapy. So on the first side, neuroprotect the system after the cyst formation, and then rebuild the damage. Today, we'll focus about neuroprotection because we're working about limiting the cascade of secondary injury using nanosystem. So coming back of what we discussed in the introduction section, among all the possible, properties that we can obtain from nanoparticle, what the property we are more interested in now is to say that to increase the cell uptake and to have a selective system that can work like Trojan horses, so enter selectively within specific cells, delivering the drug without affecting the other environment. So if we look again about which are the actors of this field, the first cells that we decided to treat more than 10 years ago, and I will go a bit quicker on that and then move to Nanogel, is based on microglia. Microglia is very important during spinal cord injury because they become macrophagic and can have a different phenotype, one good and one bad. But before to say what we said, the strategy we use to treat selective microglia is to obtain, in this case, not an nanogel that we use in the second part of this talk, but a classic polymeric micelles that self-assemble due to the presence of amphiphilic polymer, so an hydrophobic part, an hydrophilic part, and then with nanoprecipitation can create the nanoparticle. We started with the preparation of the macromonomer from the left to the right, so using hydroxyethyl methacrylate and caprolactone, ring opening polymerization to open the ring of caprolactone. In bad condition, we obtained the macromonomer. We added to this macromonomer hydroxyethyl methacrylate linked with PEG. In this case, PEG is fundamental for the stability of the system to avoid the aggregation and the instability of the colloid form at the end. Then free radical polymerization, we can obtain, as you can see on the top right, the skeleton of the polymer. This polymer presents a hydrophobic chain and hydrophilic is a comb like polymer, so they obtain, as you see from the slide, from the picture in the middle, and due to this amphiphilic nature, once nanoprecipitated in water, they create nanoparticles that self-assemble, and these are the dimension about 100 nanometers in this case. Tuning, what is important here is that tuning the amount of caprolactone, we can tune the dimension of the nanoparticle, and in particular increasing N, we can reduce the dimension of nanoparticle because we increase the hydrophobicity of the system. In this case, 100 nanometers, that for us is good. We are not, in this case, we are not very strict about the dimension. Then we added this nanoparticle to primary culture of microglia, astrocytes, and neuron. We introduced an anti-inflammatory stimulus that is lipopolysaccharide for 18 hours, and then we added the nanoparticle. We can see from the confocal picture that nanoparticle in red color because they are conjugated with rhodamine, so post-polymer functionalized with rhodamine, are present only within microglia, as you can see from the picture here, are uptaken only microglia, especially by the activated one. Is extremely clear from the confocal picture D on the bottom right, when you can see in green the membrane, in blue the nucleus of these cells, and in red all the nanoparticles uptaken. With time-lapse analysis, we also, studied the kinetics of this uptake. That is very quick, happens the first for 24 and then end after 48 hours. And considering the balance between control microglia, so not activated, and LPS activated, we can see an higher selectivity for activated microglia I would like to add. We proved the biocompatibility with the MTT and ELISA test, so that these do not affect microglia viability and cytokine release up to 6 days after the internalization. We then studied a little bit. I have to be honest, it's something that we are not very skilled on that, and we didn't, we don't have also the skills. So if someone in the audience study this aspect, is something that we are very interested in, is related to the mechanism of nanoparticle to eat, and to uptake nanoparticle. Is something that is not only completely understood by the scientific community. Most general, of course, by endocytosis, that is most common, but there are many other mechanisms that can also affect and studying specifically this aspect can, in principle, even if I told you is something that I'm looking for a collaboration around, is in principle very important in order to design the proper selective system, understanding how this uptake takes place. So basically, very, very simply, we introduce an inhibitor of endocytosis that is called promazine. So we started with primary culture microglia, astrocytes, and neuron. We obtained activated cells. Then we added chlorpromazine with different concentration that can inhibit endocytosis. What you can see from the graph on the bottom right is that increasing the concentration of chlorpromazine, you decrease the uptake of nanoparticles, but as you can see, not completely. So it means that endocytosis is very important, but it's not the only mechanism. What about the fate within the cells? These nanoparticles enter you can see from TM analysis can enter within lysosomes, and then you can see from the from the lower part of the picture that after 6 days they are almost degraded by lysosomes. We loaded an anti-inflammatory drug, minocycline, to test the ability of this nanoparticle to decrease inflammation. So you can see the group visible here are control groups, microglia itself, LPS, so microglia activated. You can see the round shape typical of activated microglia. Then you can see the two other groups are the treated group. LPS plus mino is the group where the LPS cells were treated with minocycline itself without nanocarrier, while in the other you can see the LPS with nanoparticle, so minocycline loaded within nanocarrier. You can see that this selective treatment can reduce more effectively the inflammation of the time-lapse analysis, able to go from a round-shaped structure to a spindle-shaped structure. The minocycline delivered by nanoparticle can also be selective in vivo. You can see from this injection in the spinal cord. You can see starting from the top to the bottom, the injection of nanoparticle without minocycline, with minocycline, and the last one with the nanogel, with the nanoparticle loaded with minocycline, you can see the activation. Then from the behavioral of the animals, so how the animal can better work after the injury, you can see that depending on the time of application, we can have an amelioration or not, because this depends on the drug, because as you know, in your field, it depends on the medical field, it depends on the drug and how the drug can do. Of course, we can optimize drug effect, but we cannot change. Minocycline is a standard anti-inflammatory, so it can reduce the inflammation. What is more important from this analysis is that we have results. The result depends on how strong and how efficient is the drug to do what we are asking for. We cannot, change it, but we can optimize. From in vivo fluorescent, you can see also the difference between, in terms of myelin, from the treatment of spinal cord, or this is a mouse in a mouse model, in obtain with the control group on the left and the nanoparticle loading microcycle on the right, when you can see a preserved white matter and more preserved connection and continuity in the myelin staining detected by the evaluation of fluoromyelin labeling. Then what about the main topic of this talk is the ability to target astrocytes, because microglia is a very phagocytic cells that is very, can quickly answer to foreign bodies that take place, because this is their job, you have to say. When we moved from microglia, targeting microglia to astrocyte, the level of difficulty increases, and then will increase also the next cells that you are working now. But also with hydrogen increase that much. Why? Increase that much for different kind of reasons. The first one is that astrocytes present an extremely lower phagocytic activity with respect to microglia, and also by the fact that when you have astrocytes, you have also microglia in the same environment. So what these Trojanoses should do should reduce the amount of them uptaken by microglia and move into astrocytes. Consider that microglia present an extremely higher phagocytic activity. So this is why this problem was extremely more difficult with respect to the previous one. It's commonly known by the literature, even if, as I told you, it is not is not present a real, I would like to say, a real coherence, something that is completely understood, and something where every scientist, agree that microglia are more susceptible to polymeric micelles because they present hydrophobic domain, and they, we have to say that they recognize them more like foreign bodies with respect to others. On the opposite, in order to reduce that, we said, okay, if we use something more hydrophilic, microglia can be less, these objects can be less selective. So this is why we decided to move from, because the first point is reduce the uptake of microglia, then increase the uptake in astrocyte. But the first one is reducing microglia, because if the nanoparticle enter, or all of them enter within microglia, we obtain another selective system for microglia, not for the other cells. So we decided to use nanogels, so that more hydrophilic. In this case, as you know, the main difference is that while polymeric micelles form nanoparticle through self-assembly, so it's something physical, there's not chemical reaction. In nanogel, there's a chemical reaction between bonds. So we started with PEG, so polyethylene glycol with carbonyl dimidazole, with functionalization. And the other polymer we obtained is polyethylenimine, widely used in gene delivery, that in this neat form is toxic, while cross-link not. So we introduce a post-polymer functionalization, we said before, in this case for the tracking, but it also spoiler with you that also we have to say that the chromophore is important. So we click them with a property group in the polyethylenimine and a side group in rhodamine. We obtain the polyethylenimine clicked with rhodamine. We obtain an emulsion dissolving one of them in DCM and the other in water. An emulsion using ultrasound, evaporation of the organic phase. We obtain the cross-link between the polymeric respect to physical nanoparticles, polymeric micelles, they are less efficient. This, as you know, physics is more efficient than chemistry. So you will generally obtain nanosystem with this nanogel. We introduce further polymerization strategy for two main reasons. The first one is to tune the nanogel cell interaction to a selective system, as I told you before. We introduced generally not like coating layer, but you will see that is an inappropriate, not so appropriate, not so good word to say. Or on the opposite, we'll see a few things about the possibility to use polymer functionalization to introduce cleavable linkers. What about the coating, even if it's not real coating? We are talking about very small, very, very, very small. What we did, we performed the same, the same reaction we did before. So we started with PEG, activated with imidazole. We added polyethylenimine, in this case conjugated with CY5, not rhodamine, because generally it's commonly known, and we will see if it is true or not, that the chromophore is not so important for the material properties, it's only a tracking. And basically you decide to choose you have to be clear and honest. You decide generally in the lab, I think that it is up in my lab, you decide to choose the cheapest one. So if you're in vitro, you can also use rhodamine, Fitch, and whatever. If you move to in vivo, you use CY5 and it's extremely more expensive, but basically we think that will not affect that much. And then we introduce another functionalization that is done with propargylamine. So it's not real functionalization, only we can see a very small surface decoration after that the nanogel is formed. These are how they look like in terms of dimension, 150 mostly, so a little bit bigger respect to the other. The presence of carbamate group is, from the FTIR analysis, the verification of the reaction that takes place during the formation of the nanogel that is cross-linked, so a real covalent bond, while in polymeric mice we don't have a covalent bond between the polymeric chains. And then you can see the degradation from the diameter that starts from 150 and then is reduced after 6 days. We then treated with the LPS.
Hey, Filippo, we can't hear you.
Realization in microglia. So this is this is the target that we did. So we to obtain a system, then the most of them enter within astrocyte, only few enter in microglia, and none in neuron. None in neuron is easy to perform, because even if I'm not a biologist, I know that neuron present an extremely low phagocytic. I wouldn't say no, zero, but very low phagocytic activity. So basically, they are the noble part, to say, of the system, so they're not affected by that. But basically, what we obtain is that we can have a better selectivity for astrocyte. You can see that from comparing with the other PCL nanoparticle we described in the previous part of the talk, from the graph here on the bottom right, is that you can see that the amount of PCLs of nanoparticle selective microglia that can enter within microglia is extremely higher respect to this nanogel obtained here, underlining again that what we can do is that we can reduce the uptake within microglia and then work with astrocytes. We then treated with chlorpromazine, confirming that the mechanism of uptake again of astrocyte, even if in this case is clear, because as you know from the picture AC on the up right, that using chlorpromazine, so inhibitor of endocytosis, you fluorescent indicator confirm the degradation by lysosomes again, and the colocalization of activity of confirm the degradation. Then what you can see from this analysis performed is that the amount of nanogel detected in microglia is, and you cannot see colocalization with the microglia, understand that this system can preferentially enter within astrocyte respect to microglia. Then we tested the ability of this nanosystem to carry drug within the cells and how can be done. It can be done using a molecule that itself cannot enter within cells. This is why we decided to use TOPRO3. TOPRO3 is a molecule that itself cannot cross the cell membrane. So if you can see the signal of TOPRO3 within cells, it means that the carrier carried this molecule inside. And the colocalization of the red signal of nanogels and the green of TOPO3 is confirm the encapsulation within the cells and the ability to carry them within the system. Because another topic we said that we are not highly interested on the kinetics, but we are interested that the drug is not delivered outside. So even if, when they enter, they have something to deliver. These are the results obtained in vivo, and you can see that nanogel injected in the parenchyma 3 days after the injection have internalization mostly within astrocytes, respect to microglia, and the colocalization obtained within nanogel and astrocytes is, confirmed internalization, and only the few nanogel detected in microglia and not in neurons confirm the selective internalization of astrocytes. What happened? We then loaded with rolipram, and again, now we are not at the point where we are building a real treatment, but again, to test the ability of this nanocarrier to optimize the effect of the drug. In this experiment, we loaded within nanogel rolipram. Rolipram is another anti-inflammatory drug. The difference from the graph from the left and the graph from the right is that the first one on the left, you have the nanogel injection 1 day post-injury, while in the second one on the right, 14 days post-injury. And you can see that only if you can have an amelioration at the beginning, only if you inject a nanoparticle in the first step. But we have to be honest that we don't expect a long-lasting effect because, as you know, we are working with the primary event, and then we need to add something that can rebuild the damaged tissue. So, but what we can see here is that we can give a good amelioration if in acute phase, so at the beginning, not in a sort of chronic phase where is not reducing the inflammation is not successful because there are many other events that took place, and you have to consider. In spinal cord injury, I forgot to say that acute treatment is very different from chronic treatment. What we are more interested in general, in the lab, is to work with acute phase, so before the formation of the scar, because if you work with chronic, the environment is completely different. So another strategy, another possibility, we are reaching the end of this talk, is the possibility, in order to show you all the possible things we can do, not only related to spinal cord injury. Today, I decided to focus our attention on the application we dedicated most of the time, but as I told you, my lab is involved in the material design, so also can be applied in many other field. Another possibility is related to cleavable linkers, so linkers that are not stable. Because if you load the chromophore, it should be stable, because you want to be sure that the optical signal corresponds to the particle, to the nanoparticle. So it cannot be cleaved, should not be cleaved, while if you have drug delivery, it should be cleaved in order to avoid the uncontrolled release. So an example that we did some years ago, and also we continue last year, is to perform nanogels that can load, not only load, but can be linked with the cleavable link with the drug. In this case, we decided to use a disulfide bridge. As you know, disulfide bridge are cleaved by cysteine present within the cells and then deliver the drug inside. We perform the same, we can skip a little bit, the same nanogel, and then with some post-polymer functionalization strategy, we loaded here a drug mimetic to say if they are if they can work or not. So basically, what you can see from the cumulative release on the left is that the presence of glutathione increasing the release of the molecule. So again, saying that it's able to cleave and release, in this case, the drug mimetic also from the cells. This was also looking about the delocalization of drug on nanogel in the picture on the right. We have to be honest that if this possibility to link drug is very, interesting from an academic perspective, extremely more difficult. I don't want to say impossible, and extremely more difficult if you consider about regulation and translatability, because as you know, you have to test that all the chemical reaction of the drug didn't affect the stability, the performances, and so on. Last things I would like to focus our attention, that I spoiled at the beginning, and then I promise that they are on the last few slides, is the role of chromophore. Because we notice that even if in many biomaterials, the classic biomaterial book, the role of chromophore is not is not considered. As I told you before, in like a common lab practice is that you use the cheapest chromophore, because as you know, rhodamine is extremely cheap. You have, I am sure that you are looking about the order and in the lab when you're ordering the chemical, the reagents, and you can see very highly different between rhodamine and CY5. So you won't use rhodamine, you won't use CY5 if it's not needed. But we notice that the chromophore presents a role. Which kind of role? In order to do that, we use the same nanogel before. So we started, look at the upper part. So we use PEG with CDI, polyethylene rhodamine. We obtain a nanogel that we know, we call here NG-RHB, so nanogel with rhodamine. This nanogel with rhodamine can be functionalized, decorated with propagilamine. This propagilamine created the nanogel NH2 plus RHB, so the nanogel rhodaminated with NH2. That, as I told you, rhodamine is generally used for in vitro purposes. The same can be done with CY5. So you will obtain the nanogel CY5 and the nanogel CY5 with NH2. They generally use in vivo. We so obtain these 4 nanogels with and without NH2, with or without CY5, and look about the selectivity they have. If we consider astrocyte, you can see that the presence of the chromophore partially affect the astrocyte uptake. What really, so only the presence of them, so the nature of the chromophore do not affect astrocyte uptake and visible from here. But what you can see from microglia is that in this case, you have many differences depending not only on the presence of NH2, but the presence of CY5 respect to rhodamine. If, for example, you consider nanogel with rhodamine, the first one on the left, and or respect to nanogel with CY5, you can see that the different chromophore from rhodamine and CY5 affect the microglia uptake, and CY5, the nanogel with CY5 present an extremely lower selectivity. So this is why for our purpose are extremely better respect to the other. And this coming at the end justify the choice to use NH2, because what I told you at the beginning, the first target is to reduce the selectivity of microglia and then allow the entrance within astrocyte. But the take-home message here is that not only this sort of decoration and coating influence the properties in this case in terms of selectivity, but also the chromophore, because the chromophore is a real decoration, like to say, a real functionalization on the topic. And even if it is underestimated, my suggestion is to consider is like a real ingredient in your formulation that you cannot simply change depending on the price, but depending on the final application. So basically, we are on the lab on the left. We hope to reach the clinic on the right, even if from an academic perspective, we'd like to, to think about future, because our main aim is is to publish good paper, because publishing good paper is fundamental for the growth of the science, but our final aim is to reach the patient. In order to do that, what you're trying to performing into something that can be easily reach the clinic if, of course, they have all the characteristic to reach it. And this, so I leave you with the Valley of Death, so where most of our idea die, and but I hope that most of them can pass this valley and reach the clinic in order to have an amelioration of the patient condition. So finally, I'd like to acknowledge all the people involved in this work, here from my lab, the Maroney Institute for Pharmacological Research, with the group of Dr. Veglianese, and they have an extremely strong collaboration from since 2009, and where we are dedicating our time on spinal cord injury. All the funding and the people that believed in us, and you all for your attention. Thank you very much.